It's been a few days since I did the first subculture, so the second video in this three-part series. And today I am setting up to harvest the spores from the plates, and hopefully they are from the fungus I'm interested in, Trichoderma. And I'm working in my sterile environment known as the biosafety cabinet, or BSC, and I'm putting labels on these falcon tubes that I'll use to centrifuge the spore suspension that I'll do later. I'm removing the paraffin that I used to seal it, and this is what some of the plates look like. So this is the agar that it's growing on. And I take photos for everything I work with or whenever I'm running an experiment. It's just helpful for me with my observations when I'm doing my report and my presentation. So they say a photo is worth 1,000 words, and I find that having a photo or a video helps to explain a point that I'm trying to make, because sometimes words can become too much, and if my audience has different types of learners, so it's good to have a mix of words and visual aids. I'm just going through to collect the images, and I let that play, and then I'll get back once I start my spore isolation. Something to note, something that I also look for as I take the images and label them, I'm looking for yellow-green mycelium, so the hair-like growth on the plate, the way they grow in tufts. I'm also looking for concentric circles in their growth pattern, and the smell, but that isn't very evident because it's in this environment. You see the circles here, circles like those, concentric circles, because those are part of the characteristics of this particular fungus. So Trichoderma, these are not symptoms, but these are identifiers that they have. So this is one way of morphologically identifying microbes. They have specific growth patterns on specific food type under specific conditions. This is also another growth pattern. It's not quite concentric circles, but they grow in patches as well. Note, so everything that I put into the biosafety cabinet, or BSC, the place that I'm working in, has been sprayed down with ethanol. That's what I have in that blue bottle. See, I spray my hands, and that helps to sterilize it. The inside is sterile, and I spray that down with ethanol. So now I'm setting up to harvest the spores. So I have distilled water that has been autoclaved, so boil in the giant pressure cooker. And I have a funnel, and I have a cheesecloth. So I'm just setting up my filtration or straining apparatus. This cheesecloth is also sterilized. I have a policeman. It's just a glass rod with a soft rubbery thing at the end that I use to gently dislodge the mycelium. So I'm dislodging the spores that is in the mycelium. I add a little bit of water, mix it, scrape the surface, and the spores will now come into that water suspension, and I will filter that through my 2 layers of sterile cheesecloth. I'm filtering it because I'm only interested in seeing the spores, and once I'm done, I spray my scraper with 70% ethanol to sterilize it. Because it's not a metal or glass, I don't put it in the flame, and you see it looks green, so that's what they're expecting to get. And I will sterilize my scraper in between each plate. So I think I'm going to do maybe 50 ml of water, and that's a plate without the fungus on it anymore. So that's a clean plate that will go into the biohazard bag to go into the bin. And I'll just spray my funnel to clean it up again before I move on to the other plate. The reason I'm doing that because there are many species or many types of Trichoderma, and I don't know which type I've isolated, so I didn't want to contaminate or mix them together. So that's the process. I'm scraping the surface of the plate in water, then filtering it to remove large pieces of hyphae, debris, whatever else, just so that the smaller ones that the spores will get through, and the plate becomes clean or empty, and the process continues. Spraying with ethanol to sterilize in between each plate, between each step, da 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 da. How gorgeous. Look at my soil sample. Well, not my soil sample, but the microbe, the fungus that I was interested in isolating. I think I have it, but I just added water to a colony on the agar plate to harvest the spores. And I'm going to check to see if I see the spores of the fungus I'm interested in, but it is so pretty. Wow. Now that I've isolated all of the spores I'm interested in and my samples are ready, I'm going to centrifuge them in order to separate the spores from the non-spore part. So I'm going to go into a little bit of physics, not to bore you, but the centrifuge separates materials based on the density of the material by spinning the sample at high speed, and then it creates a centrifugal force. So the centrifugal force is, as an object rotates, it will experience a force that is directed at the object from the outward towards the center of rotation. And then the density, heavier materials, so my spores, which are heavier, should fall to the bottom because they experience a greater centrifugal force. And then the stronger the force is on these denser, heavier materials, it pushes them outward towards the edge, and then that's what separates it from its liquid component. So the part that I'm interested in should fall to the bottom, and we call that a pellet, and the pellet should have my spores. Then I will decant, so that is carefully remove the liquid part or the liquid fraction from my pellet, so the little piece at the bottom, and I will resuspend the spores in water to look at them under the microscope. Usually I would gently pour off the liquid part without disturbing the pellet, but I decided not to do that in this case because this is just exploratory. So what I'm going to do is just resuspend it, right? So just put back the spores into the entire water fraction or liquid fraction, and I'm going to remove 100 microliters, so 0.1 milliliters, and place it into my little tubes, so my microcentrifuge tubes. I'm going through with the samples. Once I'm done, I'll take a smaller volume of that and put it on my microscopic slides to go see it under the microscope to see if the spores are translucent, transparent, with a yellow-greenish tint, because that is also another descriptor of what Trichoderma spores look like under the microscope. Just a heads up, I don't have any fancy camera microscope setup, so I'm using my hand to hold it against the ocular lens, so it's very shaky. But as you can see, I don't know if you can see those circular egg, they're not quite circles, they're spherical or optical, but they're yellow-green and they're translucent. So yes, it's safe to say in some of my samples, I do have Trichoderma. Yay! Woohoo! And more spores. Now I'm going to go ahead and decontaminate or sterilize my supernatant, so the remaining of the trichoderma that I throw away, I'm going to add bleach to it, and you see the color change, and I'm going to leave it until it becomes clear. So that's one way to decontaminate microbial samples using bleach. My bin with the bleach has been sitting for about an hour and a half, so now that it's clear and it's been sitting for that long, it is safe to pour down the drain. And I hope to see you next time for more adventures inside and outside of the lab, whether it's inside the lab, in my kitchen doing science in the kitchen, or on some chronicles in another country. But stick around and thank you for watching. Bye.